everyone, and welcome to our lecture on the oxidation of NADH. Here we will be talking about how we get NADH again, so just sort of recovering where the NADH is coming from. There's two sources that we've already talked about, so we'll put that together. And then we'll be looking at how we oxidize and reduce it, because remember, oxidation and reduction are paired. So oxidation is where we lose electrons, just as sort of review here, and reduction is where we gain electrons. So you're just watching the electrons bounce from one molecule to the other, and in the doing of this, we are creating energy in the end, in this case. So when we're talking about NADH and the mitochondria and all that, we are talking about getting energy here. So what we are going to be doing is essentially moving these electrons from one molecule to the other, taking them away when we oxidize NADH, and then we reduce NAD to make NADH. So we are kind of going back and forth between these, and this will allow us to essentially move the NADH from the cytoplasm into the mitochondria, because NADH is incapable of going across the outer cell membrane, or the outer mitochondrial membrane, but it is, and the inner mitochondrial membrane as well. But we are able to move what we call the reducing agent, which is just the electrons. We are able to move those across those membranes. So there are a variety of different transporters or shuttles that we will use to get those electrons across the mitochondrial membrane so that they can go into the electron transport chain, which is the whole purpose of all of this. Now, some of our NADH is coming already from the mitochondria, so we don't need to move that particular NADH because it's already in the mitochondria and already going into the electron transport chain. And that is the NADH that comes from the Krebs cycle. Now, the other source of NADH is glycolysis. That's outside of the mitochondria, so that needs to be brought into the mitochondria in order to have those electrons used by the electron transport chain. So if we are to just revisit our glycolysis metabolic map, this is an older slide from earlier in the unit, but if we look at the detail here, just a little bit hard to see, let me zoom in. We're just really looking at step 6 and 7. So here, when we turn glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate, we are coming out with NADHs. We are reducing NAD+, so adding electrons, or in the form of hydrogen in this case, to create NADH. So this is a source of NADH. And just remember that as we go from glyceraldehyde 3-phosphate and create that 1,3-biphosphoglycerate, those NADH molecules are created and they are in the cytoplasm. Okay, so that is where we are starting. We're looking at this NADH that comes from the glyceraldehyde 3-phosphate. Oops, now we can't see what I'm doing. There you go. So this is the reaction here. The glyceraldehyde 3-phosphate, so this is step 6 to 7 of glycolysis. When we do this reaction, when we look more closely at it, we see that we are adding a phosphate, and we know that when we add phosphates, we're essentially adding energy into the molecule so that it can be broken off later in order to get that energy out. So when we are doing this, we're incorporating the energy into the product here, which is 1,3-biphosphoglycerate. So that would be step 7 of glycolysis. And that is where we are getting our NAD reduced into NADH. The enzyme is glyceraldehyde 3-phosphate dehydrogenase. We can see that in our glycolysis metabolic map. What we are thinking about here, though, are two things. One, the source of NADH from the in the cytoplasm, coming out of glycolysis. And then we are thinking about adding this phosphate into the molecule that is eventually going to continue on through glycolysis. So since we're adding energy to it, we can call that an endergonic reaction. So we're adding energy in the form of phosphate into our molecule here. So if we were going to be breaking apart the phosphate and releasing the energy, that would be called an exergonic reaction. So that releases energy. And an example of that would be when you break ATP into ADP. So ATP is a 3-phosphate molecule, ADP is a 2-phosphate molecule, and in the process of doing that, you are releasing the energy, taking a phosphate off. The endergonic example of ATP and ADP would be the opposite way. If we are taking ADP and adding a phosphate to ATP, ADP plus a phosphate gives us ATP, the stored form of energy. That's another endergonic example over here. Okay, so I'm straying away from our NADH, which is what we were trying to focus on, but these are important things to just know as terminology, endergonic versus exergonic, and to know that when you are storing the phosphate into a molecule, whether it's into 1,3-biphosphoglycerate or into ADP to create ATP, that you are adding energy to it. Okay, now we need to just think about our NADH and how we're going to get those electrons into the electron transport chain so we can do this, which is the whole goal of all of this glycolysis going into the Krebs cycle, to get those electrons into the electron transport chain and to create energy. Okay, so our two mechanisms for getting our NADH, or not exactly the NADH, into the mitochondria, but getting the electrons from the NADH into the mitochondria are the glycerol phosphate shuttle and the malate aspartate shuttle. These two shuttles both work in different ways, but the goal is the same, to get those electrons across the mitochondrial membranes so that they can go into the electron transport chain. This here is just a chemical schematic of NAD plus and NADH. The only real difference is up here. Notice NADH has an extra hydrogen on it. NAD plus is missing that extra hydrogen. So when you reduce NADH or NAD plus, you get NADH. So you're adding that hydrogen with its electron onto NADH. And that is the electron that we are trying to get into the electron transport chain. So here is how we do it when we are looking at the glycerol phosphate shuttle. The glycerol phosphate shuttle is going to take the NADH that we had from glycolysis. So here's just a glycolysis in a nutshell down here. And we're taking that hydrogen, that electron, off of NADH, and we're just going to oxidize NADH here so it loses the electron. So that we have NAD plus left over. So this is still going to be in the cytoplasm out here. That NAD can then go back, since it's still in the cytoplasm, it can be used, it can be reduced again to create more NADH during glycolysis. So this will be a replenishment of our NAD plus that we can reduce in our glycolysis. Now, there's an enzyme involved in this entire reaction where we are taking another molecule called dihydroxyacetone phosphate, and we are converting it into glycerol 3-phosphate. This enzyme that does this is glycerol 3-phosphate dehydrogenase. The name right there says it all. Dehydrogenase means it's taking away or removing a hydrogen. So there's that hydrogen is being pulled off of NADH, leaving us with NAD, but it's also converting at the same time. We are converting this dihydroxyacetone phosphate into glycerol 3-phosphate. Now, glycerol 3-phosphate is able to cross the outer cell membrane, and then it is able to be worked on by the second enzyme, glycerol 3-phosphate dehydrogenase 2. So this was number 1, this is number 2. It's really just a matter of location here. But what is going to happen with this is that this removes the hydrogen from glycerol 3-phosphate, and it uses it to reduce FAD. Now FAD is in the mitochondria already. So here, this is the inner cell membrane, so this is the intermembrane space. This is the inner cell membrane that we have shown here, and the electron transport chain going from right to left instead of left to right this time. So there's complex 1, 2, 3, 4, and 5. We have our cytochrome C here, and then we have this, which is labeled here as flavoprotein dehydrogenase. Here's another example of where we have multiple names for the same thing. We have seen this before. This is coenzyme Q. It is also called ubiquinone. So if you remember from our electron transport chain, where's our electron transport chain? Hold on. In our electron transport chain, these are our notes from last time, so this is a little bit messy already. It's going from left to right this time. Complex 1, 2, 3, 4, and then 5. That would be the ATP synthase, where we actually make the ATP. So an exergonic or an endogonic reaction. But there's our ubiquinone, aka coenzyme Q. So this is where we are going to be dealing with our FAD and our FADH, being as ubiquinone is able to transfer those hydrogens or those electrons from FADH2 to FAD and vice versa. So this is where we are creating FADH2, where we can use those electrons in order to continue bouncing down the electron transport chain. So this is just showing what we already know or what we've seen, but just how we're getting those electrons from outside the mitochondria inside the mitochondria and then allowing them to enter. So the NADH never crosses the mitochondrial membrane. All it's doing is donating its electrons in order to reduce a series of molecules so that we can transfer those electrons to FAD at the ubiquinone part of the electron transport chain. So right on that inner cell membrane, or inner mitochondrial membrane, and then we'll have FADH2. And there we can go through the electron transport chain. So in a nutshell, the process is that electrons from NADH plus, or NADH, are moved to glycerol 3-phosphate, and then through ubiquinone to FAD to form FADH2, and then those electrons can continue on down the electron transport chain. Okay, now in the meantime, as we have taken those electrons from an NADH and moved them onto glycerol 3-phosphate into the intermembrane space, then the ubiquinone takes those electrons, adds them to FAD to form FADH2, and then on through the electron transport chain. In the process, we have created NAD+ out here in the cytoplasm and FAD in the matrix of the mitochondria. So that FAD can then be reduced, if that makes sense. We can reduce it again into FADH2. So we've replenished our supply of NAD in the cytoplasm, we replenish our supply of FAD. Really, those things are just used as almost intermediaries to carry those electrons from one place to another. Okay. Okay, so this slide is just a reiteration of the previous slide, just giving all the details here listed in bullet form. So just reminding you that the NAD+ is going to stay in the cytoplasm. We can turn it into NADH again. The next time we do glycolysis, which is pretty much constant. The electrons from NADH, which is now NAD, are going to be transferred into this glycerol 3-phosphate and then into ubiquinone. So here it's calling it flavoprotein dehydrogenase again, just to use the other names for it, so that you just are aware that we use multiple names for the same thing, just to make this more complicated. But the ubiquinone is then going to reduce FAD. It says it uses it as a cofactor. We're reducing the FAD into FADH2. Now, one thing we didn't mention in the previous slide is that glycerol 3-phosphate, that is how we're carrying those electrons, but we kind of need this dihydroxyacetone phosphate in order to create the glycerol 3-phosphate while taking the electrons off of NADH. So we have to resupply this in order to continue to do this entire process, to make a perfect little circle here. And that's what we're going to do is, after we've taken the electrons away, the same enzyme here, the same ubiquinone protein that has got this enzyme embedded in this inner mitochondrial membrane, is going to just reverse the reaction and turn that glycerol 3-phosphate back into dihydroxyacetone phosphate, and we can just do it all over again. So we're going to see this in our next shuttle system as well, where we have this nice little circle of, we have one product, we have the dihydroxyacetone phosphate turning into glycerol 3-phosphate. And in the meanwhile, while we do that, that is causing us to take our hydrogen off of NADH and creating our NAD again and sending that hydrogen on into the electron transport chain in the end. It goes there in the end. And then we will just simply reverse that reaction so that we can just keep doing it over and over and over again, because we're going to keep having a steady supply, hopefully, of NADH that we need to turn into NAD and grab the hydrogen. So we need to just keep doing that over and over again. Okay, so our next shuttle that we're going to talk about is the malate aspartate shuttle. Just another way of getting the hydrogens or the electrons from the NADH into the mitochondria. So we'll pause this video here, we'll stop this video, and we'll do a separate video for the malate aspartate shuttle.